The chapter 6 slides just got put up like 20 minutes ago. So if you didn't see them, they are there now. I'll get the rest of the unit slides up hopefully later on this week. So you'll have those to look at if you want to see them before, print them off or anything like that. Yeah. So like I said, chapter 6 kind of starts off. We're going to get like a pinch into physics and chemistry, but not too far. Yes. Yeah. Yeah, and they should be, they've been letting me know when people do one-on-ones. That's actually, I can go ahead and, yeah, everybody can know about that. So there are the snap groups, and I've been giving points for every week you attend some form of a snap group. But yeah, if either the schedule doesn't work for you or you need one-on-one tutoring or virtual tutoring, you know, there's other stuff that goes on. Madison and Cassie are letting me know when people are meeting for that too. So that also counts. I'm not, you know, if you're spending time studying, then I'm like for it. So yeah. Yes. Oh, uh-huh. Oh, okay. Yeah. No worries. Yeah, and those will get added. When I grade your exams, I'll probably just write your snap. If you've got snap points added, I'll add whatever you've got to the exam when you see it. Sure. Trying to think. Other things. Quiz, lecture journal, makeup exam, grading exam. Yeah, okay. Other questions? Anything else going on? All right. Chapter 6. So chapter 6, again, is kind of just, we're gonna, I say briefly, it's superficially pick apart some physics terms and things we need to kind of move forward into respiration and photosynthesis. So we'll start with kinetic and potential energy, right? Talking about kind of the forms of energy, how we see it used and stored. Talk about where that energy is coming from when we're talking about living systems, and also revisit redox reactions because they're going to be coming up a lot in basically every process we're going to be unpacking for the next little while. So just to make sure we all kind of remember what's going on there, right? So to start, what is energy? Energy is just, for our purposes, the capacity to do work, right? Energy is used to do work. So however much energy you have, are using, are storing, right, represents your ability or capacity to do something. And it can exist in two kind of states, lots of forms, but two states, right? Either potential or kinetic. So potential is just the potential to do work, right? It's stored energy. So like a stretched rubber band, or in the case of all of these metabolic pathways, covalent bonds, right? We've talked about covalent bonds storing energy, right? It's a form, it is energy that is stored, it is potential energy, right? And when we actually use that energy, release the rubber band or break the chemical bond, right? It becomes kinetic energy, or energy in motion, energy that is actively doing something, right? So that's easy enough, right? So energy is either kind of waiting to be used or it is being used. It's kind of in one of those two spaces at any given time, right? Is doing or is not doing. Right? So it exists in these two states, but it can be in a lot of forms. We don't always kind of think about energy as being in so many forms, but you know, some of them seem kind of logical, some of them seem a little counterintuitive, but like heat and electricity, sound, radioactivity, right, are all kind of forms of energy, right? Kind of all things that can do work. For our purposes, because it's both kind of simpler to think about and it is just kind of the energy that our internal systems are using, we're going to focus on heat. It's the most convenient for a couple different reasons, right? So heat, right, kind of technically separate from temperature, heat is the measure of random movement of molecules. Now, the more molecules are moving, the higher the temperature is. So they're very related, right? But heat is specifically a form of energy, and it's one that comes into play a lot in biological systems. It's also kind of easily measurable in the big scheme of things, right? This is what calories are. Calories, like in the food that you eat, are measurements of heat, right? In the case of food, it's the measure of the heat as potential energy, if that makes sense, right? So when we talk about calories, kind of our go-to unit is actually a kilocalorie. Remember your units of measure from lab 1 if you're in the lab currently, right? Kilocalorie is 1,000 calories, specifically 1,000 lowercase c calories. I don't know who made this kind of system, but it is a little confusing. It wasn't my idea, right? But lowercase calories, 1,000 calories, right? So 1,000 calories equals 1 kilocalorie. 1 lowercase c calorie is the heat required to raise 1 gram of water 1 degree Celsius. That's a phrase we've heard before. What is that? I've said that phrase a few times. It is. It's the specific heat of water, right? Specific heat was the amount of heat needed to raise 1 gram of substance 1 degree Celsius. And so in the case of calories, we're actually talking specifically about the specific heat of water, which kind of makes sense. All our biological stuff is in this kind of aqueous system in one form or another, right? So 1 calorie is how much heat, energy you need to raise 1 gram of water 1 degree Celsius. And in the form that we tend to see it in our daily lives, 1 kilocalorie, lowercase c, is equal to 1 capital C calorie, right? And that 1 capital C calorie is the one that goes on food labels, right? So your food, the energy stored in the food that you're eating is basically recorded in kilocalories. And for short, they just write capital C. Yep. Yeah, so if you see like a large C on calorie, it means kilocalorie. So it's actually 1,000 little calories. Yeah, like I don't know whose bright idea that was. It's not confusing at all, but it wasn't me. Right, so we can measure stored energy in calories, which is a measurement of energy or heat specifically. Right, and that energy ultimately, right, the whole biosphere, we're not a closed system, right? The planet is not a closed system. We actually have a constant, large input of energy, huge, right? In the form of light and heat energy coming from the sun, right? And so we're not necessarily directly able to use this heat, but there are organisms, right, autotrophs, that can harness this energy coming from the sun and convert it into other forms that then we're able to use, right? So things that photosynthesize, plants, algae, a lot of bacteria, right, are able to capture that heat and light energy coming into Earth from the sun and convert it into forms of energy that kind of the rest of life on Earth is able to use, right? And it's actually kind of cool if you think such things are cool, right? So photosynthesis is using this energy from the sun, combining it with carbon dioxide and water, and turning that into more complicated molecules in the form of sugars, right? Generally glucose, right, is kind of the end of photosynthesis as we tend to discuss it, which is really cool because they're doing something that none of us can do. They're taking inorganic compounds and converting them to organic compounds, which both they and the rest of us need to exist, right? And so metabolically, this is just something that like our bodies can't do. We can't convert these inorganic resources to organic ones to store them as energy, but photosynthesizers can, right? So that's kind of the chain of things happening. We've got sun coming into our autotrophs, and then by consuming autotrophs or other things that consume autotrophs, you basically take in kind of second or third or fourth hand that heat energy that was coming in from the sun. Right? And so that energy is being stored in the covalent bonds in things like sugars, lipids, carbs, etc. Just conceptually kind of a bizarre thing to think about, I think. All right. Real quick. All the way back to week 2 for just a second. We're going to review redox reactions. You may have them. You may be on top of redox reactions. If not, let's look at them real quick again. Right, so a redox reaction is an oxidation and a reduction reaction, oxidation being an atom or a molecule losing an electron, right? Remember this goofy little picture, right? So we've got molecular atom with an electron, and the electron bugs off. That's oxidation, right? Something loses an electron. Reduction is just the opposite of that. Some atom or molecule gains an electron that it encounters bopping about in space, right? So oxidation, we lose an electron. Reduction, we gain an electron. Generally speaking, and particularly in biological systems, these reactions always they always happen in pairs, right? Because if you're in a system and some atom loses an electron, you've got this little charged particle. It's going to stick to something. So invariably, some other atom or molecule is going to pick up that electron. Right? So oxidation happens. Pretty much always reduction happens shortly thereafter. So we just refer to these collectively, right? We just call them redox reactions. Because if one is happening, the other is happening. And these reactions are actually really key in energy flow through systems, right? As these electrons kind of pop on and pop off of things in systems, they're moving energy through biological systems. So we'll see redox reactions happening quite a lot. So just to kind of make sure those terms stay a bit fresh. All right. Briefly, a bit more physicsy, chemophysicsy, I guess. So we're going to talk a bit about thermodynamics, which is kind of where chemistry and physics meet, right? We're going to talk about energy changes in chemical reactions, right? And then add enzymes into that whole process. So let's do that. Right. So again, thermodynamics, right? We talked about the very beginning of the semester. Biology is kind of at the convergence of chemistry and physics, right? Physics acting on generally organic molecules where we're interested, right? And the results of all of those interactions is what kind of causes biological systems to function the way that we see them function, right? Thermodynamics specifically is the branch of chemistry that is concerned with energy changes, right? So looking at changes in energy across reactions. And if metabolism is creating and using energy, right, then this is going to come into play. So first law of thermodynamics, energy is never created or destroyed, right? We've heard these before. People say them a lot, right? So there's energy in the system. You can use that energy. You can change it. You can't make it, and you cannot destroy it. You can lose it. It can go somewhere else. It can change forms. It can become something that's no use to you, but you can't destroy it, right? We can move things from potential to kinetic, right? Change things from electric to heat, right? You can change the form, you can change the state, but you don't actually destroy it or create it, right? We've got constant input. We're not creating it, but the sun's putting it into the system constantly. And we're kind of using it. And as we use it, we tend to lose a bunch of it. We're not very efficient, turns out, right? But we can't create it or destroy it, right? So in living systems, we're storing all of this chemical potential energy that we've converted from this initial heat energy from the sun. We store it in molecules, right? All those carbon-hydrogen bonds that are holding energy that our bodies are able to use. And then we can convert that into kinetic energy that we use to move and do all the things we do, right? And this is going to kind of direct us towards the second law of thermodynamics, because in every conversion, every time you change it from one thing to another, you lose a little bit. We're never 100% efficient in any of these processes, right? But basically, if you consume something, you are consuming the potential energy in the covalent bonds of the thing you're consuming. That's what eating is doing, which is a really weird kind of way to think about it, right? But this heat energy was put into organic molecules, covalent bonds. You eat those covalent bonds, and your body breaks those bonds to use as energy. That is what is happening in your body all the time. Now, second law, All these reactions are happening. We're losing energy all the time. Energy is lost as disorder increases. This is that idea of entropy or kind of chaos in the universe, right? In a natural kind of closed system, things will just go kind of towards disorder, right? Things will just kind of bop about and fall around and lose energy and change things, right? Because we can't make these changes completely efficiently. So anytime things are happening, you're kind of losing energy. Things are happening a little less efficiently than you might like, right? So this comes to that idea of entropy. And entropy is just kind of that idea that disorder is more likely than order, right? If you don't input any extra energy, little dude chucking bricks here, right? If you throw bricks, they're going to fall in a pile. They're not going to fall on a wall unless you add more energy into making them be ordered, right? It costs you to make things ordered, right? So the converse of that is kind of the natural state is disorder, is a way to think about it, right? So energy is lost as disorder increases. So that's our second law, right? Again, unless you add more energy to make it ordered, in which case you're still losing energy, right? Ice changing to water is this sort of thing, right? If you put ice kind of in a natural system, it will naturally become water, which is technically disordered, right? You've got water, which is a crystal. It will become less ordered as it turns to water. Anyway, all right. So we can't create or destroy energy. Things will naturally kind of become more disordered if we don't do anything to stop them from becoming disordered. Another concept we want to kind of pick up is this idea of free energy. So free energy is just the energy available to do work, to break and reform chemical bonds for our purposes, right? Chemical bonds increase order, right? The more energy we store in bonds, we're making kind of an ordered system. Adding heat, breaking bonds reduces order. Free energy is whatever kind of energy is available to break and reform bonds, right? To store energy as potential energy and to break a bond and release energy as kinetic energy, most likely. Increase order. Creating a chemical bond increases order because you're kind of holding the energy in place. It's kind of a weird way to think about it, but it works. For our part, we're just going to say heat reduces order for now. Adding heat will reduce order, right? Because you increase energy, things start moving faster and become slightly less ordered. So you have these two kind of plays between temperature and the bonds present. Okay. So in a system, the energy that you have available to do work is called free energy. It's the energy free for doing work. That makes sense. All right. So how do we know how much free energy we have? We're not actually going to do math in this class, but we're going to look at an equation that will help us think about how our body's using energy, right? So when we want to know how much energy we've got available, there's a couple of things that we want to take into account. Enthalpy and this term TS that we'll talk about in a second. Enthalpy is that potential energy in the chemical bonds, right? So enthalpy is this potential energy in the molecules that we're looking at. We're going to call it H when we look at our equation. Enthalpy, we're going to call it H, and it's just how much energy do we have available to us in the bonds in some molecules, right? So that's kind of our order, right? Chemical bonds are order. We also need to take into account that kind of idea of entropy or disorder. It's going to kind of eat up some of the energy we've got available, which is going to be the TS term. So it's another energy term, and it's again related to the disorder. So right, so if all the bonds that we have present are kind of our order, right, our potential energy that's all tidy and packed away, TS is going to account for the entropy, for the disorder that we're going to have to kind of fight back against to do work, right? And so those two things we talked about, right? Heat or temperature, and then the entropy or disorder that is present, right, are going to be things we're going to have to kind of fight back against to keep the system ordered. That makes sense. So we've got this H term and this term TS. All right. Now the difference in basically your order and your disorder is going to be equal to your free energy. We're using Gibbs free energy. We're not going to talk about Gibbs or the history of that. Just the letter in the equation is going to be G. That's what that gets us. So free energy represented as G in the equation makes it look like this. So if G is the energy I have available, it is equal to the energy I have available kind of minus this entropy, right? Minus this disorder that's going to kind of reduce the amount of energy I actually have available. All right, so my order minus my disorder, sounds really strange, equals the total amount of energy I've got available to do stuff. All right, so that's great. It means you can look at a molecule, you can look at the bonds in it and say these bonds hold this much energy, but given the temperature and the kind of disorder around the molecule, I've really only got G, this much energy left, which is great, but it's not really useful just to be like, this glucose has this much free energy. Well, that's great. Who cares, right? We're looking at chemical reactions, right? Metabolism are like long series of chemical reactions. So what we're really interested in is the change, right? So when these chemical reactions happen, we're breaking bonds in some reactants. We're going to make new bonds in the products. And so we're looking at changes in free energy, right? Because that H term, right? The bonds on the reactant side that make that H term are going to be different on the product side because there's different bonds present, right? And so this leads to a change in free energy, right? If we look at the reactant side and the product side, the free energy here will be different than the free energy on the other side. Okay. Why do I care? I don't know. We're going to just assume that temperature, pressure, and volume stay constant, right? Theoretically, your cells are in some sort of form of homeostasis. Your temperature stays constant. Hopefully, the pressures and volumes of your cells aren't changing too wildly. That would be problematic. So we can get rid of our T and then just tack some deltas on here, basically, right? Delta just means the difference in. So the change in free energy is going to be equal to the change in bonds between the products and reactants minus the change in entropy between the products and reactants. Okay, cool. So why do I care? Because the change in free energy can tell us whether a reaction is spontaneous or not, right? Are two molecules in your body going to interact or not? If we need them to, what do we need to do to make a reaction happen, right? So this change in free energy, the energy of the products that we end up with, minus the energy of the reactants that we started with is the difference, right? Between the starting and ending energy levels. Starting and ending free energy, or delta G. It's just delta G, that's all that is, right? So if we look at delta G, we can decide whether or not a reaction will happen spontaneously. Cool. So if it's positive, right? If I subtract, so if this is my free energy, right? This is my ending free energy. If I subtract my beginning free energy and the number is positive, then the products have more free energy than the reactants. Cool. All right, so this means that in my products, the bond energy here is higher, or maybe the disorder is lower, right? This is more organized, it's holding more energy. But it also means that to get from the reactants to the products, we need an input of energy, right? It's an uphill reaction to get from one side to the other, right? And so this reaction is not going to be spontaneous, right? Where it's going to take energy to get these reactants to make these specific products. And the term for that is endergonic. Right. So if delta G is positive, right, if our products have a higher free energy than our reactants, then it is not spontaneous. It is an endergonic reaction. Energy has to be supplied to those reactants to get them to become those products, basically. Conversely, if delta G is negative, right? If I have free energy of my products minus the free energy of my reactants and I get a negative number, right? My products have less free energy. The bond energy is lower. Maybe there's more disorder. And so this reaction, when it happens, is going to release energy. It's a downhill reaction. Yeah. And so because we're going to release energy, we don't need energy necessarily to make it happen. That's going to be a spontaneous reaction. Because our reactants have more free energy than the products. We call that an exergonic reaction. So if you have kind of high free energy reactants making low free energy products, all the extra energy is actually going to be released. We'll talk a bit more about this later. This is one way that your body's able to do work, by doing these reactions that are going to release energy, right? That will then become kinetic, right? We'll move from potential to kinetic and be able to do work. All right, now, just to add another layer on here, chemical reactions are reversible, right? We always write kind of chemical reactions from left to right, makes sense in our heads, and we just kind of talk about them going from products to reactants. But really what's happening if you mix reactants in a beaker or whatever is that the chemical reaction is actually going back and forth, right? Everything's kind of reacting together and going back and forth. So if you have a reaction that is, say, endergonic, uphill in one direction, right? When it's coming back the other way, the other direction, it's going to be exergonic, right? So any chemical reaction is going to be spontaneous in one direction and non-spontaneous in the other direction. So they're kind of going back and forth.
So is spontaneous, exergonic is not spontaneous, or is the opposite?
Opposite.
So it's not spontaneous. It's not spontaneous or whatever that word.
Endergonic is not spontaneous. Exergonic is spontaneous. Yeah. So if every chemical reaction is kind of going back and forth between products and reactants, I mean, it doesn't necessarily happen like kind of for infinity or whatever, right? There's going to be some point of equilibrium where things kind of balance out. And that's called the equilibrium constant, right? In every chemical reaction, it's kind of moving between reactants and products, and there's going to be a point where it finds balance between kind of the relative amount of reactants and the relative amount of products. It's going to depend on these changes in free energy between the two sides of the reaction. So the exergonic spontaneous reaction will favor the products, right? So if this reaction going from reactants to products is downhill, right? As this is going back and forth, it's going to end up over here. You're going to end up with more products, right? Because it's easier to go this way. So this will favor the products. If you're looking at an endergonic, a non-spontaneous reaction, kind of left to its own devices, it's going to favor the reactants, right? Because this is uphill. Unless something kind of comes in and gives us some energy, we're going to be mostly stuck over here. We're not going to do much reacting, right? Because it's a non-spontaneous reaction. So exergonic reactions will have an equilibrium that favors the products, and an endergonic or non-spontaneous reaction will favor the reactants. Right? Basically, they're going to kind of favor their downhill side, right? That makes sense. It takes less energy to be there. All right, now, most reactions, even if they're spontaneous, need a little kind of nudge to get going. the energy it takes to kind of start a reaction is called the activation energy. So even if a reaction once it started was going to spontaneously just keep happening, we frequently need kind of a little input of energy just to kind of like get the ball rolling, right? And that's the activation energy for a reaction. All right, so the extra energy that you need to kind of destabilize the bonds in a molecule to make a reaction start happening is called activation energy. Now, if it's endergonic, if it's non-spontaneous, right, we're going to need a large input of energy. We kind of already covered that. But if it's exergonic, if it's spontaneous, there is still kind of a need for this activation energy. But it may just be the tiniest little bit, right? It may not be a lot. And so the rate of an exergonic reaction will end up depending on how much activation energy it needs to get going, at least in part. Right? If it just needs like a tiny little nudge to get going, then the reaction will start really easily and probably go really quickly. If you have a reaction that's spontaneous, but it needs kind of a heftier push to get it going, then the reaction rate is going to be a bit slower, right? So the more activation energy you need, the lower the reaction rate, right? The slower the reaction is going to proceed because you need to kind of give it this big push at the beginning. So this means that generally speaking, we can increase the rate of reaction in two ways. We can add energy. We can increase the energy of the reacting molecules, right? Because that was one thing. Some reactions just need an input of energy. Or we can lower the activation energy we need to start a reaction. Right, so in the case of increasing energy, right, we talked about this, I think we mentioned it briefly, right? You can add heat. Things like industrial processes frequently add heat to reactions they need to happen to make them happen more quickly. So we can add heat, or we can lower the activation energy by adding a catalyst, right? So if we have a reaction that needs a chunk of activation energy, we can add a catalyst to kind of handle that for us, basically. Right? So catalysis is stressing bonds to lower activation energy, right? And so this affects kind of the transition state of a reaction. Right, whether we're kind of over here or over here. We can kind of start destabilizing bonds and make things move from one side to the other, right? So if you have a reaction that's exergonic, overall it's heading downhill, but we kind of needs like a hefty push to get things started. We can add a catalyst that will kind of shake up these bonds to begin with, and we can get going a little bit quicker, right? Now these catalysts work both forward and back, kind of in the same amount. So they're kind of destabilizing bonds on both sides. They're not particular to the products or reactants, right? All they're doing are kind of destabilizing the bonds present. And so the direction of the reaction, right, whether equilibrium has us end up with more products or more reactants still depends on the change in free energy. We'll just get there quicker if we catalyze the reaction. All right, in living systems, this is done by enzymes, right? So those proteins that are able to speed up biological reactions as needed, right? So we'll spend some time talking about enzymes probably Wednesday. All right, probably finish up with this section. So we're going to talk a bit about ATP specifically and where it kind of comes into all of this exergonic, endergonic reaction, making things happen situation, right? Because we know that ATP powers things. We've seen that already. But how exactly does it do it? So it's a nucleotide. Remember we were talking about all those biological molecules. We talked about DNA and RNA are made of nucleotides, but there's also these kind of other nucleotides that do some different sort of things, right? ATP, adenosine triphosphate, is one of those. It's got three main parts, right? So we've got a 5-carbon sugar. We've got a little ribose attached to an adenine. And then we've got this 3-phosphate chain, right? Phosphate is just phosphorus kind of surrounded in oxygen. So we've got these 3 phosphates all chained together, right? The sugar just kind of forms the framework, right? It's kind of the backbone that the other pieces are attached to, right? Not critical in the sense that it's needed structurally, but it's not super active. Neither is the adenine. The adenine, right, is this kind of organic molecule. It's 2 of these kind of carbon-nitrogen rings. It's just a weak base. But the part that we're really concerned with, the part that like will be doing things that we're paying attention to in the ATP is that phosphate chain, right? So we've got this carbon sugar, we've got this adenine, and then this phosphate chain. And this 3-phosphate chain is highly, highly negatively charged. And so because each of the phosphates is highly negatively charged, they repel each other, right? It's like holding kind of the wrong sides of magnets together. They're actually pushing hard against each other. It gives this kind of coiled spring sort of characteristic to this phosphate chain, right? And so these bonds between these phosphates are kind of, they're the active part of this. They're the part that we're kind of more interested in, right, when we're talking about ATP, right? So because these phosphates are negatively charged, because they're actually, even though they're bonded, they're already pushing against each other, they're going to have a super low activation energy, right? They basically don't want to be bonded together functionally. And so hydrolysis reactions, right, adding a little bit of water is going to easily break this bond. It'll happen really fast, and it's going to transfer energy, right? When you break that bond, the bond is holding a lot of potential energy, and that energy is going to be released, right? So in breaking these phosphate bonds, right, here, these high energy bonds, we can release energy. Right? In other words, using the terms we've been using, the hydrolysis of ATP, breaking this bond, has a negative change in free energy. It is a very steeply downhill reaction, highly exergonic. And so the released energy is then, right, the released free energy is then free energy available to do work, which is what ATP does, right? We use it to power other things. All right. Generally speaking, we'll see a couple exceptions as we go along, but generally speaking, only the outermost phosphate is hydrolyzed, right? So we add water, we break this bond, right? And what we're left with, right, if we remove this phosphate, we're left with an adenosine diphosphate, right? Because there's 2 phosphates on it now. A phosphate group, and then the energy we've created, right, is the result of that reaction. So we break off a phosphate group. We've got that kind of inorganic phosphate hanging around, this diphosphate, and then a bunch of free energy. Right? Yep, hit both buttons. Right, so ATP, once it loses that phosphate, becomes ADP. And the phosphate is generally just stuck to whatever we were applying this energy to. It's kind of the way it works. We break off that phosphate and it sticks to kind of whatever we were powering. We saw this in the sodium potassium pump. If you remember, we attached ATP, it got dephosphorylated, and that little phosphate group stayed stuck to that protein, right? So we powered the protein, changed its shape, we left that little phosphate stuck there, and the ADP went back to wherever it came from. Right, so the phosphate, so the energy is used to do work. The ADP goes back to be turned into another ATP, and the phosphate stays stuck to whatever molecule we were trying to get to do something to begin with. So this is how ATP powers non-spontaneous reactions. It comes in, we add a little water, we break that phosphate bond, and the energy that's released is enough to catalyze, or to, well, not to catalyze, to start reactions that would not otherwise happen on their own, right? They make up for those uphill reactions, right? We can add enough energy to overcome the uphillness. All right. Like I kind of alluded to a second ago, ATP is used cyclically, right? So exergonic reactions, spontaneous reactions, provide energy that is used to make ATP from these ADPs and phosphates that are kind of hanging out in the cell. So we're doing reactions that give us energy. We attach phosphates to ADP, and then we send the ATP, right? Because we've got diphosphate, you add a phosphate, it's triphosphate. That adenosine triphosphate is then sent somewhere where it's needed, hydrolyzed, and releases its free energy for whatever process we need to make happen. Right. And so your body, all your cells are doing this constantly. All the things that need ATP to run, they're basically breaking ATP and sending it back to get another phosphate, and breaking it again and sending it back to get another phosphate, just over and over and over, right? So that at any point really in your cells, you actually only have a few seconds worth of ATP. It's just being constantly and quickly regenerated in each of your cells. Yeah. Right. So we've got ATP to power things, right? Our other option is to use enzymes. And so when we come in on Wednesday, we're going to start with enzymes, right? And finish out chapter 6, most likely on Wednesday. Yeah, so that's all I've got right now. Yeah, so we're going to stop at ATP. We'll pick up enzymes on Wednesday. If you need the makeup exam, again, make sure that you're in this hallway at 11 a.m. tomorrow.